Good afternoon, and thank you for joining us today. So let me first thank the organizers for inviting me to give this talk, especially Michel Taranto. So I'm going to discuss or explain the physics of the face. So I've put here two very beautiful individuals, Alain Delon and Audrey Hepburn. So, of course, the face is related to the individual. It's something very personal. And physics is the science of the general laws. So you may believe that there might not be a general law for faces, but I'm going to explain you that there has been recent progress in morphogenesis, especially in developmental biology of embryos. And there has been progress in the description of the formation of the body, and also more recently in the description of the formation of the face, so that there exists a science of the face, and each individual is one instance of a general law. So the face is an interesting question, maybe not problem, but question from a human point of view, because of course it's related to psychological issues, to our sensitivity, to our connection with others. And independently of psychology, the face is related to our identity, since a identity photography is a portrait, and for your passport, you will take an identity photo, it's a portrait. Now, it raises interesting questions in embryology. How does a face form in an embryo? And that may lead to teratology questions, babies which might be born with malformations of the face. And that will take you, or might take you, to medicine and surgery, because if you want to reconstruct the face, either because of a newborn malformation or because of an accident, then likely you will be interested in knowing very precisely how the face is structured. Now, there are, of course, evolutionary and paleontological issues related to the change of face during evolution between, for example, fish, birds, primates, humans. So all these animals have faces which are slightly different, but nevertheless, they do have a face. So how do faces change during evolution? And can we have a hint into how faces evolve up to humans? Now, of course, all this is related to genetics. Here to the right, I've taken a picture on internet. So some people are very fond of putting their faces on internet with their parent. So here we have a mother and a daughter. And of course, the game is to try to guess who is who. And my guess is that here to the right, the red-haired girl is or woman is the mother, and probably she is the daughter. So if you go to biology papers or textbooks, you'll find a description of sensory organs formation, such that, okay, in the blastula, at a very early stage, when the embryo is still more or less flat, you have cranial placodes. And these placodes, after some process of induction, will become the sensory organs, like the olfactory pit, the optic vesicle, the otic pit, and some other things like a trigeminal nerves and things like that. And you will find plates showing the evolution or the development of the animal or of human in utero, and showing different stages. So this kind of plate, you find them on internet or in textbooks. They represent head formation at different stages, and you see that there are folds, cavities, involution of tubes, of nasal pits. Now, all this is very precise. It's very organized. It's sort of flexible because the embryo changes all the time. And of course, not two individuals are identical and not two animal species are alike. So there is a flexibility. But still, these plates are not quite clear because they don't show the principle of formation. Why don't they show you in the first place the principle of formation? So maybe it's not understood, or maybe there is no principle of formation. This is a movie that was shown a few years ago by the BBC. It's generated by assembling pictures of embryos at different stages. And the result is the visual impression of a time-lapse movie of development. But it's a bit misleading because each image is a different embryo. And so the movements that they reconstructed are not the actual movements in vivo. So this is the traditional view of embryo development. It's called the Carnegie stages of human development. And you see that at this stage, for example, here, you have a recognizable baby. But if you ask, okay, when does the embryo form? Okay, probably here, you have a complete embryo which has rudiments of everything, rudiments of eye, limbs, lungs, organs, brain, etc. At this stage, most animals are identical, almost indistinguishable. So the formation of the rudiment of animal, of the larva, so to speak, occurs very early between a state in which nothing is present and a state where almost everything is present. So there is a very rapid phenomenon that sort of generates the overall structure of the embryo. Of course, this is not completely accurate because human embryos develop in the bag of waters. And so you should add the chorion and the placenta, which are embryonic organs, actually extra embryonic organs, but they are made by the embryo. So these pictures are dissected embryos, and part of the embryonic tissue is missing, this other part. So we are not allowed to work with human embryos. We work mostly with chicken embryos. And so I'm going to explain you the development of the chicken up to more or less here. And then I will describe in more detail the formation of the face. So my talk will be divided into two parts, and I will shift from one PowerPoint to the other. So this is different snapshots of chicken head formation. So you see that there is a rough, not formed stuff that sort of folds, and you see the extension of the eye stalk that forms eye capsule, which becomes properly an eye. And after a couple of days, maybe 1 day and a half or 2 days, you get a recognizable face. So it's not clear whether there are actually stages. It's more like a constant deformation of a fluid-like stuff. So our goal today is to understand the general mechanism of head formation. And instead of relying on snapshots, we will rely on time-lapse movies. So I'm going to show you a lot of time-lapse videos. Unfortunately, the compression algorithms, they spoil a little bit the images. So the images, the original movies are much nicer and crispier than what will appear on the final MP4 movie. So this is a lamprey. It's called the flute fish in English. The flute fish, and this is a Alain Delon. So how come you can transform during evolution something like a primitive fish into a human? Now, nevertheless, you see that the primitive fish is very simple. It has a shaft, a main body, which is roughly a cylinder. And at second order, there are details like the mouth, which is round, the eyes, which are round, and a series of holes which are cylindrical. So this looks almost like an artifact. It's a very simple thing. So how come that after 600 million years of evolution, you still find in the seas such a simple animal? So is biology that complex after all? So I'm going to take a physical approach to explain this simplicity. So the physical approach is based on a biomechanical description of animal formation. And I expect that if you are interested in tensegrity, then you are interested in forces, pressure, tension, and so you are acquainted somewhat with physics. But I will assume that you really don't know deeply the physics, and I will go very pedestrian-like. So I'm going to explain the formation of the body, the formation of the head, and you may wonder whether it's arbitrary. You know, we give names to parts, but biology does not give names to parts. It's not conceivable to make a head without a body or a body without a head. So maybe the distinction between body and head is arbitrary, and we'll see whether it's arbitrary or not, what is the difference between the sensory organs and the rest of the body. From a physicist point of view, if you want to address the problem physically, you need to find a reference configuration, the initial state. In fact, as you will see, the formation of an embryo is some sort like a viscous flow. It's like a paste which becomes an embryo. So where do you start? So you need a reference configuration. So for me, the reference configuration will be early stage of oocyte cleavage. And I will not address the origin of life or the origin of sexuality or the origin of chromosomes. I will address the mechanics of how a mass transforms into an embryo. Now, this mass, what sort of stuff is it? So in physics, we speak of the constitutive law of the material. You may have viscous materials, fluids. You may have solids, elastic solids, hyperelastic solids. You may have other forms of material like crystal liquids. So what sort of stuff is it? So at first order, it's a viscoelastic gel. So in fact, embryos are more or less like marmalade, like a jellyfish. Now, once you know the reference configuration and the kind of stuff, you need to know why does it deform? So if it deforms, it's because there are forces inside it. So what are the forces acting in the mass? What is pulling, what is pushing, etc. So once you have found 1, 2, 3, the thing unfolds. So the scenario of development unfolds all by itself. And if you are lucky enough, there will be an attractor of the dynamics. And the attractor of the dynamics is what you will call the animal. So there is no randomness, no hazard at all in the phenomenon of development, or women make children automatically. But certainly there is some randomness in the initial parameters of the problem. But once you have chosen the parameters of the problem, then the things unfolds all by itself. So of course, in order to do the biomechanical approach, you have to skip, to leapfrog, to bypass the biology. So if this is the tree of life, you have a lot of randomness in the evolution of animals, mostly due to mutations in connection to the biotope. And so the biotope is just sort of random, and that selects animal and modifies the parameters. I'm not going to discuss that. During the developmental process, you have all genes which are making positive and negative feedbacks on each other, regulating, downregulating transcription factors. So people try to address and find all the relationships between genes, one cell inside a single cell, the dialogue between cells. I'm not going to discuss that either. And at the molecular level, of course, you have proteins interactions, like ligand interactions or antigenic interactions. And so this is all the proteomics. I'm not going to discuss either the energetics of protein folding and interaction. Still, we can address most of the development without knowing anything about the biology if you have the proper hypothesis. So recent progress has managed to understand body formation on a simple set of hypotheses. First of all, the initial configuration for development is much simpler than expected. The distribution of forces is much simpler than expected, and the stuff is simpler than expected. So roughly, the stuff of which the embryo is made is a gel, slightly liquid crystal-like, but it's a gel. And the reference configuration, the starting geometry, it's a disc which has rings inside, like 3 rings. And the forces, the embryo is making itself, it's not made from the outside. So if the structure of the cells is in rings, and since the forces are exerted by cells, then forces also are in rings. So in fact, inside the embryo, there are belts of cells which pull, which may become strings or cables. And so if you have a disc of gel, which is pulled by rings of forces, the attractor is a fish, a chicken, or whatever, okay? So this is a chicken in the first day of development, and you see by eyeball that there are rings on the developing blastula. This is a blastula dissected off from a very early stage of chicken development, and you see that there is an external ring, there is an internal ring, and there is a more internal disc. And when you go and magnify and properly film the structure, you see that the central part, which is going to give the nerves, is made of small cells. The next ring is made of bigger cells. The next ring is made of even bigger cells. And the peripheral ring is made of even bigger cells. So there is a very regular structure inside the embryo. It's made of rings with a stepwise variation of cell sizes. So at early stages, the structure is organized in rings. This is a chicken. You have big cells at the periphery, medium-sized cells, and then smaller cells. So it's like that. Now, a chicken is birds are sort of special. I mean, birds have a flat blastula up to the point that they call it a blastodisc, while other animals, here is a frog, they are more of a spherical blastula or morula, and you recognize the rings here. It's a profile view. This is a top view. This is a profile view. There are small cells here, medium cells, larger cells, okay? Human, they are in between the frogs and chicken, and mice have a more barrel-like shape, but there are just slight differences which will not interest us today. So during development, you have cleavages at constant volume. So you're maybe not aware of that, but the first divisions during embryo formation occur at a constant volume. And it's a matter of cleaving this mass. And you have a first cleavage, a second cleavage. And after a while, you get a ball with an internal structure, sort of tiling. And as I've just explained, the tiling has a structure which resembles a Hanoi Tower, you know, this game of Hanoi Tower. You have these plates of small cells, medium cells, bigger cells. So these are small, a few rounds of division when the egg is inside the hen. So this is from the work of Lee and Al. And so this is a chicken, and you see little by little, a structure in rings stabilizes itself, much like 3 rings. Now, why are cells smaller in the center and bigger at the periphery? It's because the yolk has an inhibiting effect on cell division. So cell division is very slow at the periphery, while it's very rapid in the center. Okay, so this is why cells are smaller in the center they divide more. Then cells at the periphery, they divide less. And so you have a stepwise variation of cells, which corresponds to cells which are in the same round of division. Okay. So now that you reach this situation by day 1 of development, this is a blastula seen by magnification 4x under a microscope. So this is the edge. So at this scale, the blastula is slightly bigger than my screen. And what happens is that it starts to rotate. Okay. So there is a rotation like that of the blastula. And there is a posterior movement and an anterior movement. And the movement starts by the edge and it rotates inwardly. Okay. Now, this is going to be the anus. This is the anal area. So the movement is towards the anal area and away. And the little white spots here are the nuclei. So if you superimpose the plates, you do a Z projection of the stack of images in the movie. Then you see these little dots, they are the nuclei. And then you see perfectly well, you have large vortices, which are fluid vortices. It's a fluid flow. And there is a stagnation point here of 0 speed. The flow goes away in that direction and converges in that direction. We call it a saddle point. And so the anus is going to be the saddle point, or rather the saddle point is going to be the anus. This is the posterior area, and this is the presumptive anterior area. So this is a fluid flow. And why is there such a fluid flow? Is because one of the rings, the peripheral ring, is the one which starts to constrict first. So there is a force by the edge. The ring pulls from the two sides, it constricts, and that generates this viscous rotation in a very thin disk of gel. It's a physical problem. Now, this rotation continues, and now there is an invagination in the hole that I may explain why it occurs, but so to speak, you have a collision, a frontal shock of all the cells, and they pack up, and they can escape to the top in the vortices, but they can also do a small inwards rotation, and then they flow underneath. So they flow underneath. So you see here the underneath flow. And meanwhile they flow, they continue pulling, and the continuous pull ends up in a very strong anterior-posterior pull that forms a folds on the embryo. So it's like pulling on a rubber sheet. Okay, it tends to fold. So you can model that with simple fluid flow models. So this is a very simple, exact mathematical model. If you have a contraction like this, that generates a flow inside the blastodisc, and you can do a more refined calculation in which you generate a viscous flow in a plate by pulling along a line. What you need is a sluggish viscous flow, which is called a Stokes flow in physics, a term of tension along the ring. But now I add something. The tension is not uniform, because if the tension would be uniform, the thing would really just implode. You need a gradient so that the tension is stronger by the anus. So there is a maximum of tension at the pole. And to generate this kind of image, you need just a cosine 1 minus sinus theta function. So the tension value is 2 here and 0 here. There is a gradient, and this is why the flow is oriented towards the anus. And the origin of that is in the fact that the division of the cells are slightly skewed from anterior to posterior. So cells are bigger posteriorly, so they pull a little bit more. This is an actual map of speeds in blastula. So you have this invagination of the jelly through the anus, okay, a very beautiful lava-like flow. And by the end, there is this pull, which is strong enough to fold the surface. If it wouldn't be strong enough to fold the surface, then it would remain flat. But it just happens that the values of the parameters are such that when I pull, okay, you need a certain threshold to fold the surface. And here the threshold is exceeded. So you rotate towards the anus, you invaginate part of the stuff, then you pull and it starts to fold, okay? So we are dealing with traction forces which generate active folds, and that form the body. So if we follow this traction, you see that over a few hours, it's very rapid, it takes maybe 6 hours, you generate a embryo of embryo, a rudiment of embryo, which is some sort of larva, which has a rudiment of eyes, rudiment of brain, the few rudiments of vertebrae, which are called somites. Here are the chicks, rudiment of chicks, and this is a neural tube. So you see that it's a viscoelastic problem of folds. This is a dorsal view, and this is a ventral view. Okay, you have the cavity here, which is going to be filled up with the esophagus, stomach, and gut, etc. Now, this process continues, and it continues by extended caudally until closure of the anus. And while it extends, it generates all the somites, maybe tens of somites, up to hundreds in snakes. I don't know how many vertebrae we have, maybe 30 or 40, but snakes may have up to 300. So this is a movie in the posterior part. You see the formation of the spine. And this is more in the anterior part. You see the formation of the brain and of the eye precursors, but we're going to go into more detail. So it's a folding wave, okay? So you can superimpose the plates and see the streamlines of the flow. But the important fact is that if you magnify this area, you see the bigger cells. Here you see the medium-sized cells, and in the nervous area, you see the smallest cells. So in fact, these rings that were initially present, they deform during the formation of the embryo and the folding process, but they don't mix. The area of bigger cells, the area of medium cells, the area of small cells remain neighbors, but independent neighbors, and the folds occur exactly, okay, these folds, they occur exactly at the boundaries between these domains. So the boundaries pre-pattern the territories by locking the folds. So the boundaries between cells, this is the place where folds occur, okay, but the folds are, so to speak, locked to these boundaries. It's much like when you have a sleeve, okay, the sleeve, you have the wrist and you have the arm, and you will always find a fold at the boundary, a very specific boundary between the wrist and the arm of the sleeve. And this occurs because of the mismatch in elasticity. So during the rotatory movement and the propagation of the flow and of the folds, you see that here it's hardly fold. It's the image is blurred because the fold is beginning, so it's slightly out of plane. But you see that to the left, on the flank, the cells are large, medium-sized, and to the inside the presumptive neural tube, the cells are small, and the fold folds exactly along the boundary. But in fact, if you take an embryo which has not folded yet and you fold it by brute force, by pulling on it, then you see that the embryo folds at a specific boundary which is latent, okay? And as I've told you before, here you don't see why it folds there, but it folds there because you have bigger cells on one side and smaller cells on the other side. So what you can do is you pull on the embryo and you see that the embryo is stiff in the center, it doesn't deform much, and soft laterally. So in fact, the areas where cells are bigger is soft, and the area where cells are smaller is stiff, okay? So you can do a cross section of the embryo, you pull on it, and you see that there is a sharp variation at some point, which is the boundary between the small cells and the big cells. And you see that when you pull on the embryo, it deforms not a lot in the central part, and it deforms much more in the lateral part. This is also a color coding of the deformation of the embryo, and you see it deforms more by the edge than centrally. So this means that the rings, central part and the rings, they have different elasticity. Okay, and you see that there is a shoulder or a step of a variation of what? This is a cell size as measured really by counting the cells and the diameter of the cells. You see you have large cells here, small cells here, large cells here, small, and there is a boundary between small and large cells. So in fact, the embryo, it's like an elastic rubber foil with softer and stiffer areas, which falls into nested tubes like Russian dolls. So if you think of this rubber sheet, okay, then you would need to put a tag, okay? You would put a tag in the center of the embryo, so you would end up with a situation like that. You have a stiff area close inside a softer area, and so when you pull on that, it's not easy to do live, but when you pull on that, okay, the folds, okay, the folds are sort of, to speak, refracted. I mean, they follow the edge of the stiffer area. And this is how you generate a stiff tube surrounded by a softer tube. But it's tubes inside tubes, like Russian dolls. Okay. So in fact, the reference configuration is made of rings. The rings pull, and the result of the pull is an animal whose structure is a tube, inside tubes, inside tubes. Nested rings ends up into nested tubes. And that's a unique phenomenon. But birds are amniotes, so they develop inside the bag. And so far, there was no bag. Now, you see here that the central disc has become the neural tube. The neural tube has an apex, which is the tail. And what happens is that the tail scratches the surface, and there, it triggers a contraction of just another ring. And the contraction of this another ring generates a bag. Okay, so this is on the caudal side, and this is on the cranial side. Okay, and both rings converge, coalesce, and that ends up in the complete closure, okay, of the bag over the baby. And this is the exact moment when the belt of cells here constricts and wraps the baby. Okay. Now, if you go closer and earlier, and you see that along the extra-embryonic region, there is a belt of cells, and these cells, they contain filaments. They are fibroblastic-like, just like muscle cells. And in fact, the blastula is just starting to do exercise. You know, the embryos, they do sports. But if you wind back the development of the embryos, at all stages, they do sports. And even when the embryo is flat, it starts to exercise. And the exercise that the blastula makes is the developmental process. Once you are made, you do sports. But when you are a blastula, making sport consists in making the baby. Okay. So at this stage here, everything is flat, but cells are starting to cooperate and to pull along this belt. And after some time, the belt condenses, becomes stronger, and the thing becomes completely 3D, and it becomes a purse. You have a string that constricts, and it becomes a bag and a purse. This is an underneath view, and this is a top view. This is why the optical grain is not the same in these two movies. So what you see is that you have aligned cells which form strings or cables. The forces co-localize with the steps of elasticity. And so the dynamics, which is the pulling force, comes together with the geometry. These are two faces of the same token. The geometry is in rings, and the forces are in rings too. Now, if you look in more detail, the movie, you scratch here, that constricts, but this ring also constricts. Here, there is another ring that we just forgot, which is the boundary between the dorsal side and the ventral side of the body. And that constricts and folds along the circle to generate the dorsal and the ventral part of the embryo. Okay, so you have, in fact, more rings than expected. Along these rings, you have pulling forces, which are represented by the dashed lines. And self-consistently, the geometry changes under its own force. And so the rings self-deform autonomously into an embryo, which is a tube inside a tube inside a tube. But there is more tubes, which is the chorion, which is wrapping the embryo. All right. This is a SEM, scanning electron micrography, in which you see the string that forms the amniotic sac. The spine has become a very elongated tube. And here you have the future limb, which is starting to fold. So it's exactly, it's an embryo which is frozen exactly there, okay? Now, if you go magnify more, you see that there is a ventral ectoderm, a dorsal ectoderm, and there is indeed a difference in cells, with smaller cells here and bigger cells there. So the folds follow the boundaries between cell types. If you magnify more the string, the cable, you see that it's tendon-like, it's like a little muscle. So in fact, the forces that drive the development of the embryos is just the same as the muscle force. And the cells are muscle-like. So schematically, you have rings that are already deformed into a very elongated ellipsoid tube. You have more rings which are flat, and the tail scratches the surface and causes the contraction of one ring, which becomes the limb, and another ring, which becomes the amniotic sac. So this is the schematics of the formation of the body. All right. Now, of course, you feel like asking, what exactly is this force? And the molecular origin of this force is lies in actin-myosin filaments. So you have filaments inside the cells, and these filaments, it's a difference with plants, have the property that they can pull on each other. And you have a head of myosin, which can make a conformational change under the presence of ATP and of calcium ions. And in the presence of calcium ions and ATP, then the head will pull the filament. And this is how the force is exerted. Okay. So I will not go into this detail, but the only thing we need is filaments that are actively pulling. And that suffices to generate rings that pull. Okay. So now I'm going to shift to the next part, which is to explain the formation of the face on this tube.